The reason actually any of this matters is because when we use this approach, when we restore NKT function with our therapies, not only does it remove senescent cells quite quickly, but that alone resolves different diseases, both metabolic diseases and fibrotic diseases, as we've shown to date.
Hello, everybody. My name is Jose Navarro Betancourt, and I am the scientific director of Quadroscope. We invest in early stage healthcare companies that enable the reversal of biological age in order to treat and prevent aging and age-related diseases. And today, I will be having a conversation with Robin Mansukhani, the co-founder and the CEO of Decidius Therapeutics, one of the companies in our portfolio tackling one of the hallmarks of aging known as cellular senescence. Robin, thank you again for taking the time to talk to me.
Thank you, Jose. Thanks for having me on.
So, why don't we begin with a brief overview of the company and the diseases that you're focused on treating?
Yeah, sure. So, the unique angle that Deciduous is taking around senescence is we did the work to understand how the immune system naturally regulates senescent cells. So, said differently, when you are healthy, your immune system does a great job of selectively and consistently removing your pathological senescent cells that develop from, age, disease, injuries, natural parts of biological aging in different organs. What happens over time is that your immune system, in this case, particularly the natural killer T cells, which are a very small, often overlooked T cell population of immune cells, they don't function as well with age and in the presence of disease. And so what happens is they stop removing senescent cells as effectively as they could when you were younger and healthier and disease-free. And so what we've done that's really unique is we've actually figured out that it was in fact natural killer T cells that regulate this process. That was part of our founding mission. But the reason actually any of this matters is because when we use this approach, when we restore NKT function with our therapies, not only does it remove senescent cells quite quickly, but that alone resolves different diseases, both metabolic diseases and fibrotic diseases, as we've shown to date. So that's really why it's important.
And what sparked your interest in longevity healthcare in general, and in particular cellular senescence? We touched
Um,
on that senescence can treat multiple diseases, but why
yeah.
senescence in particular?
Yeah, I just thought, one, the logic is very strong, right? So you have these pathological senescent cells that shouldn't be there for very long, but they stick around, and obviously they're sticking around. They're not having a positive impact when they're very inflammatory and pathological. Um, but the scientific reasoning is really that it surprised me early. I started looking at senescence, 7 or 8 years ago, way before even thinking about deciduous. It really surprised me that just removing this one type of pathological cell, these senescent cells, could not just slow disease or halt disease, but in many cases we were seeing a reversal in disease. And so I just thought logically, this is a cell, type of cell that should not be persisting in the body. And when different approaches can be used to remove them, that you can potentially reverse different diseases of aging. And I thought that opportunity was going to be quite significant.
So, in particular, you're focusing on treating pulmonary fibrosis and
Mm.
type 2 diabetes.
Yeah.
Why are these diseases a good model or a suitable disease to be treated with senescence in particular? Given that
Yeah.
you given that you can pick many other diseases, why
Yeah.
those?
Yes, I would say that it's kind of in your question. There are many things you could go after with senescence removal technology. So it's not by randomness that we chose pulmonary fibrosis and diabetes, but it is where we started. There's many more places we can go. Part of the rationale in pulmonary fibrosis and diabetes is that in humans, senescent cells are shown to be causal in those diseases. So you think about translation between mouse and human. One question that a pessimist would always ask is, when you remove stem cells in a mouse, how does that line up with what it would look like in a human? And it so happens that in both pulmonary fibrosis and in type 2 diabetes, there's a very strong correlation between senescence accumulation in disease severity in those two diseases. And so that's why it's a good place to think about applying your technology. Um, More so than that, what we've shown is in these indications, when we remove senescent cells in a pulmonary fibrosis model, we can resolve fibrosis by about 75 to 80%. And when we do it in a high fat diet type 2 diabetes model, we can also make those animals um, metabolically regular once again from a single dose. So the impact of the treatment in both those models we've shown to be in preclinically really quite significant. And if they were to translate one to one from the mouse to the human, it would represent a revolution in both those indications.
Indeed, it would be a true revolution in the treatment. So I think at this point it would be helpful to make a brief parenthesis into what is a senescent cell.
Yep.
Uh, so initially, a senescent cell was defined as a cell that stopped dividing, that we call this an irreversible cell cycle arrest. And instead of functioning normally, it produces a bunch of growth factors and inflammatory molecules that damage the tissue where it lives and the whole organism in general.
Yep.
So having this type of cells as a treatment target gave rise to two strategies. The first one that we touched on is senolytics, which
Yep.
is let's destroy these cells. And then the second one is the xenomorphics, which is like to reverse the senescence phenotype or
Mhm.
to produce less damaging factors into the environment. Can we maybe explain the rationale to why senolytics became perhaps a more popular strategy than xenomorphics?
Yeah, they're different strategies, and I would say the senolytic field is much more is much further along. It's an older field than xenomorphics. Xenomorphics may have some potential. Um, it just isn't as nearly as far along as the senolytic field. So within the senolytic field, one thing I want to point out is that the initial targets that people were going after were wrought with significant challenges with regards to safety. So, for example, early targets in senolytics tended to focus on the anti-apoptotic pathways. And so if I just want to unpack that for a moment, right? So your cells when they become senescent are becoming irreversibly damaged and going into permanent cell cycle arrest. And when they go into permanent cell cycle arrest, they start to upregulate an anti-apoptotic pathway. And so basically a fancy way of saying they're overexpressing a pathway that prevents that senescent cell from being killed. And that's problematic. And so the initial therapeutic strategy had been, okay, if they're expressing this anti-apoptotic pathway, which could be something like BCL2 or P53 or P16 or P21 pathways, let's go. They're overexpressing those pathways. Let's put in a drug that inhibits those pathways, right? So if you inhibit those pathways on a senescent cell, then in theory, that senescent cell is no longer in an anti-apoptotic state and can now be killed. The downside with that approach has been that P16, P21, P53, BCL2, even beta-gal, which are all these anti-apoptotic targets, they exist. Those pathways are overexpressed in not just senescent cells, but also in healthy cells. So it's not specific to a senescent cell. They do exist in senescent cells, but they also exist in healthy cells like neurons, beta cells, macrophages, etc. So the early senolytic field, was focused on these pathways, thinking more, in my opinion, about the efficacy with it while not paying attention to the off-target safety side effects that would ultimately doom a lot of these programs. So that's where the field has struggled to date. Now there are ways to make a better senolytic and a better anti-apoptotic target that's more specific, but none of those are going to be perfect. Um, the immune approach we have obviously takes all into account, just says, let's just reboot the immune system and do what it should have been doing to begin with. And that's why our approach, I think works so well and why the safety results we have so far are also quite promising. The xenomorphic approach is really around, these cells are, instead of removing the senescent cells, can we just dampen their effects? That's the idea. Now the issue is that sometimes, it's hard to target those senescent cells and just xenomorphic profiles. They can be some can be specific towards senescent cells, some can be non-specific. Also, some of that inflammatory profile that the senescent cells are giving off are also trying to signal danger to the immune system. And so if you and basically it's kind of like waving a flare in the middle of the night and saying, "Hey, there's a problem here." And so if you put a drug against just some of the inflammatory components coming out of a senescent cell, you might be missing the bigger picture that there's actually a problem in that organ or that tissue that needs to be addressed. And so you might just be dampening the effect while basically cloaking it, and that would result in a inability then to understand what's happening or the immune system to react properly to a organ or tissue in need. So that's where I think the xenomorphic challenge, I think that's what needs to be proven by that group.
Thank you. That's a very nice overview. So trying to summarize, the first generation of xenotherapeutics that is actually currently in clinical trials or even available as supplements has faced some challenges in terms of the dosing, in terms of the selectivity
Yep.
for senescent cells, particularly because they are focusing on anti-apoptotic pathways that actually healthy cells sometimes need them as
Yep.
well as senescent cells. So that really limits the doses. And maybe a quick parenthesis here. This lack of selectivity is one of the reasons that many companies working on senolytics have chosen to look for indications or diseases where the treatment can be administered locally, right? Like in the eye, in the retina, or in the
Yep.
joints, or for osteoarthritis, or even in the skin. We touched on another kind of controversial topic in senolytics. You mentioned beta-gal, which is a marker of senescent cells.
Yep.
What other markers can we use for senescent cells? What's your
Yeah.
take on identifying senescent cells?
Yeah, the problem is there's really no single marker that's going to give you a definitive answer. The way that we do is we actually gate. So we look at beta gal cells. So let me just back up. So to answer your question, there are other markers: P16, P21, P53, MDM2, BCL2, all control anti-apoptotic pathways that do link to senescence. But as I said before, also can be present in very normal, productive, healthy cells. Um, I think the best way that we can do it is by gating it. So not just having a single marker, but having multiple markers and having it pass through each filter. So, for example, within the beta gal markers, there's something called beta gal high and beta gal low. And what that means, it's basically just a rudimentary way of saying beta gal high basically means more inflammatory, more of the SASP or cytokine profile you'd expect with a senescent cell. Beta gal low means that we think it's senescent, it's expressing beta gal, but it's not expressing any of the inflammatory cytokines commonly associated with a senescent cell. So to recap, beta gal low means, possibly senescent, but not expressing a lot of the inflammatory phenotype. Beta gal high would mean, expressing a number, very high number of the SAS genes or SAS markers commonly associated with senescent cells. So that's one way to just say, okay, we can get a more precise read on which cells are senescent and pathological versus which ones are non-senescent, or maybe senescent but not yet pathological. That's one way we do it. There's further gates and markers we use for this to weed out, non-senescent cells. And so ultimately, when we look at senescent cells, we're actually measuring what we call our beta gal high cells, which are commonly expressing the SASP profile. And so that gives you basically a look into tens of different cytokine markers that would be commonly associated with senescent cells and gives you a more accurate read on what you're actually, which cell types you're actually impacting.
And by gating, you mean using flow cytometry, correct?
We use flow, and then within that population, immune cells can exist too. So we don't want to look at the immune cells, these macrophage immune cells, and so we remove that from the view because those would give you a false positive.
And for a bit more context, the cool thing about flow cytometry and the whole gating strategy, create gates for specific cell populations,
Yep.
is that you can have very complex panels.
Yes.
right? You can look at beta gal and many other surface proteins or even intracellular proteins. That's the power of flow cytometry and this
Yeah.
kind of instrument. Let's dive a little bit into monitoring the efficacy of these treatments. Now, even if you have a very neat gating strategy and a very good way to identify senescent cells, how would you tailor the doses when you don't have tissue samples available? For example, when treating a person, right?
Yeah.
Getting a lung biopsy wouldn't be that simple. For example, when you have a disease, you can monitor the progression of the disease and say,
Yeah.
hey, the treatment is working and it's healing the disease or it's slowing down the progression. But if you want to use the treatment as a preventive or preventative intervention,
Mhm.
what do you measure?
Yeah, preventative I think is to come in the future. I don't think we go there yet. Preventative, it's a different bar. You really got to show your safety profile is very, very clean and the efficacy is also quite good before you go preventative. Um, in order to understand the dosing, it's really about just doing a dose ranging study in the clinic, right? So you can do a SAD or MAD study, single ascending dose or multiple ascending dose, and that'll tell you, what's the upper bound on what patient compliance is, meaning safe, what patient tolerability. And two, what's the dose at which you get a nice efficacy readout? And the efficacy readout really, you can do biopsies of fat. So if we look at type 2 diabetes, you get biopsy of the fat and look at NKT cell engagement and expansion in actual patient tissues and patient samples. But ultimately, what you really care about, we gotta, senescence is really interesting as a mechanism, but ultimately, what you really, really care about is does it actually treat disease in a way that's quantitative and irrefutable, right? A lot of these, some of these studies that we do in the clinic, have qualitative readouts like patient-reported outcomes, like PROs. And what's challenging with that is they become very noisy and become, they're highly variable and can be very heterogeneous. When you have quantitative readouts, like you can get in diabetes or you can get them in IPF, it just tells you without anyone's opinion or anyone's reporting that this thing is working or not. And they're all disease specific, So for example, in diabetes, you look at HbA1c levels after 1 month, after 2 months, after 3 months, and is it going down or not? You look at insulin levels, you look at fasting blood glucose levels, glucose tolerance levels. Are they improving or are they not? In lung fibrosis, you look at, forced vital capacity, FVC, and this is basically how much air you can push out of your lungs. It's a quantitative measure. And so there's less variability driven by patients, and it's more quantitative. And I think that ultimately that's what's going to tell you, does all this translate or not?
That all makes sense. In the end, what you want is to heal something, at least in the first stage of drug development.
Yeah.
Now, let's dive into some emerging evidence that senescence has physiologic roles in normal development
Yeah.
and normal function, right? So there's a series of new research showing that clearing senescent cells can actually delay uh, tissue healing or even promote fibrosis. There's also some evidence looking at the role of senescence in the placenta and in the
Yeah.
development of the placenta. So how are you addressing these emerging evidence and these positive potential effects
Yeah.
of senescence?
Yeah, it kind of goes back to what we talked about 10 minutes ago. It's really about, do we actually understand what a senescent cell is?
Hmm.
Right. There are senescent cells that are non-pathological that have a really nice role, really important role in embryogenesis and in wound healing. And that's well known. But they tend not to be pathological senescent cells. They tend to just, it's almost like they're in some ways they probably just shouldn't even be called senescent cells because their inflammatory profile is just so different than one that I would tell you is a pathological senescent cell. So I think that's part of the challenge with senolytics and one of the things with the immune approach we have, it really only focuses on the targets on inflammatory pathological senescent cells because it's an immune cell, so it's picking up inflammation. It's reading the inflammation from the cells and going after those ones. So those are, there are positive impacts that are known about senescent cells, but they tend not to be the pathological ones. They tend to be very acute. But embryogenesis and wound healing tend to be two areas where, um, in my opinion, it's a misnomer, but where, these non-pathological senescent cells have a positive impact. And so I think that's that. So I think the field we just need to understand and define senescent cells um, in not just one cell type and not talking about as one single type of cell, but understand that there are different types of these cells, and some are bad and some are good. And the good ones tend to be in the areas I mentioned and tend not to be very inflammatory, pathological, whereas the bad ones tend to be the ones that are expressing or overexpressing the cytokines related to inflammation.
And another helpful way of understanding this might be comparing it with inflammation, right?
Yes.
Inflammation, acute inflammation is good because it helps repair the injury,
Yep.
right? And that's what these senescent cells might be doing. But chronic inflammation, on the other hand, it's a low-grade pathological state. We touched at the beginning of our conversation on the immune system, and it seems that one of the uh, processes that the immune system does regarding senescent cells is that once the senescent cell fulfills its purpose after an injury, the immune system has to wipe them out, right? It has to destroy them.
Yep.
How does the immune system identify the senescent cells that need to be destroyed?
Yeah. So it gets a little bit into our novel compound, so I'll give a kind of a high level surface answer first. So what we found is that senescent cells overexpress CD1D. And it turns out that NKT cells exclusively recognize CD1D. So pathological senescent cells, say it again, pathological senescent cells overexpress CD1D.
So
And
sorry for the interruption, but when you say overexpressed CD1D, this is a protein in the surface of the cells,
yes.
right? So this means that they present it outside the cell, and so another immune system cell an immune system cell can recognize it and destroy it.
Yeah,
Uh,
but the thing is only NKT cells recognize CD1D. So that's where the discovery came about, is that you have it's just a very high fidelity immune marriage between CD1D and NKT cells. They only really talk to each other. And so when you have a cells overexpressing CD1D on its surface, the NKT cell is the only immune cell type that can really recognize or um, would be interested in that overexpression. So that's where it initially comes from. Now there's more to around the actual killing and other receptors and ligands that the NKT cell is looking for in the senescent cell, but that's at least at a surface level, it's CD1D driven.
Maybe we should dive into how your company is targeting these senescent cells involved in aging via this CD1D surface marker.
Yeah, so it's very straightforward actually. So the NKT cells are being targeted by a novel drug we made,
Mm-hmm.
and the drug has been designed to fit into a receptor on the NKT cell. And when it fits into that receptor, it activates and proliferates the NKT cell. And what happens is the NKT cell, when it's activated and proliferated, there's basically they become more effective, and there's more of them. And when they're more effective and there's more of them, they can then recognize the senescent cells that are overexpressing CD1D in the tissue. So what's happening is the NKT cells are being activated and proliferated in the tissue of interest. And when they become proliferated and active again, they will then basically wake up and do their job to remove the senescent cells overexpressing CD1D on the surface. And so that takes about 4 days from the injection happens. The NKT cells need time to activate and proliferate. And once they do, they start removing the senescent cells in the tissue very quickly. So we see that within 4 days, we can activate, proliferate the cells, and cause removal of senescent cells all in that 4 day window.
And how permanent is this effect?
Yeah.
Will the NK
Yeah.
cells keep on removing new senescent cells? How often do you have to like remind them to attack the
Yeah.
cells?
Yeah. So in a mouse and a human, it's going to be a little bit different. What we've shown so far is that one dose will give us about 3 months of efficacy in a disease model. And so when I say in a disease model, it means we have a mouse and we're giving it constantly the injury, in this case, a high fat diet. and we give that diet every single day to the mouse. We're not letting up on the animal to see if it can just naturally resolve when we stop injuring it or giving high fat diets. We actually push the model very hard where we say, okay, we're going to keep these mice on high fat diets the entire time. We're going to treat with our drug during that time, and we're going to see, in the intersection of our treatment versus the animal being on the high fat diet, how long will this last? And it turns out it's around 3 months. In a human, I'd expect it to be even less because humans accumulate senescent cells much more slowly than even mice. And so if you have a drug that's effective in removing them, you shouldn't have to give these drugs very frequently at all, but that will be ultimately determined in a clinical study. If I had to guess as we sit here today, I'd say at most every 6 months, more likely, depending on disease, probably once a year is what you'll see the treatment regimen being.
That makes sense. This is what some people in the field call hit and run approach.
Yeah. Yeah, we
Because
call it intermittent dosing, but yeah, hit and run is also another way to say it.
yeah, intermittent dosing is more
Yeah.
appropriate, more formal.
Yes.
Um, so when do you anticipate to begin clinical trials if everything goes well?
Yeah, so we're scaling the compound right now, finalizing the formulation. We've run one safety study already in rats, so it's just at this point it's selecting the final formulation, and then running a number of different safety studies in both small and large animals, both IND enabling, and we still have a little bit more of pre-IND work left to do on the non-GLP side. So just, I think it's just those steps. Unfortunately, those studies can take a little bit of time. So they're about 4 to 6 months per study. And so we'd anticipate clinical studies happening sometime in 2026.
2026. So would it be appropriate to kind of describe your treatment as a vaccine for senescent cells that kind of stimulates the immune system to attack them?
Yeah, I think it's a fair way to say that. I mean, think of it as a vaccine regimen where you're giving, basically you're just priming the immune system to be ready for, in this case, for senescent cells. And so, yeah, I think a vaccine regimen and that connotation is applicable here.
Very cool. And talking about engineering receptors of immune system cells,
Yep.
in this case with a small molecule. But another approach could be engineering the receptor genetically, right? There are some reports of CAR T cells
Yeah.
being engineered in order to attack senescent cells. And there's newer technology to edit NK cells, the CAR NK cells. Do you
Yeah.
see any challenges or opportunities on using CAR NK cells?
Yeah, I think certainly the research phase is a very viable approach. I think I'd be curious about the specificity of those and how quickly, or how well they can specifically target a real pathological senescent cell. So I think there's still questions to be answered around specificity and will it have any off-target effects. And then two, probably the bigger challenge is, with any CAR approach, the manufacturing, the scalability, the cost, um, and the translation for safety into humans can be pretty challenging. We've seen that over and over again with other CAR approaches, especially in cancer, where the preclinical efficacy looks very strong, but when you get into humans, manufacturing and safety becomes um, quite a challenge. So I think that's where there's still things to be determined on the CAR NK or CAR or different CAR approaches for senescence. Um, and so that would be my concern with those approaches. It's not to say they can't work, just say that it's an extra layer of complexity, extra layers of complexity with cost, safety, and manufacturing to get them to work. So if you can have something that's less expensive, easier to manufacture, and is known to have proper specificity, that will always be the winner.
In, Indeed, that's a fair comparison.
Yeah.
Cell therapies are still very expensive in
Yep.
the order of the hundreds of thousands of dollars,
Yep.
while small molecules are cheaper to manufacture.
Yeah. Hundreds of dollars per dose versus tens of thousands.
Um, so Are there any upcoming or ongoing clinical trials of xenotherapeutics that you would recommend us to follow up closely? For example, any cool progress in the field lately?
Yeah, I think Unity's made some good progress with their BCL2 inhibitor for DME. It's shown a single dose of that drug with it's given directly into the eye, so that delivery is not ideal, but it does have seem to have an impact in a number of patients on their vision. So like I said, it's good to have these kind of irrefutable quantitative readouts. And so I think that's been a promising study to watch so far. We know that in DME you get BCL2 positive cells that are senescent in the eye, and that's so this drug would work against removing those would work on removing those BCL2 positive cells in the eye that are likely to be senescent. And so some of the clinical data that's come out so far from the single dosage has shown that vision does improve in a number of patients over about the course of a year. So that's promising. I mean, think about that treatment regimen: one dose, get about a 1 year improvement in vision. Not all patients responded, to that, but a fair number of them did. So it seems to have potential in the field. And it just shows the utility that if you can, it just scratches the surface so that if you can remove senescent cells, even if it's not in a non-ideal ROA, like through the eye, but there can be improvements in phenotype, and that's what we're all after.
And yeah, that's a great example. It's actually, if I remember correctly, one of uh, the mechanism of action of these treatments is inhibiting the anti-apoptotic
Yes.
mechanism, right? It's
Yeah.
one of the first generation of
Yeah.
senolytics.
The BCL2 pathway actually came about from a drug called navitoclax, which is made by AbbVie, and so it's a cancer drug actually. So they reprogrammed that cancer drug to also work on senescent cells. So there's some good there, there's some challenges there with anytime you reprogram cancer drugs to work on senescence, there are obviously going to be questions, but that's what it's based on. And so there is some press in the field for BCL2 inhibition being effective in cancer, and now it's being applied to senescence.
Another company that I would like to highlight is perhaps Rubedo Life Sciences.
Yeah.
They were in the news recently. They raised $40 million to advance their program into clinical trials. I believe they're looking at skin conditions. They're looking at atopic dermatitis and psoriasis.
Yeah.
What should we be paying attention to in this trial?
Yeah, I think going for skin first is a very interesting approach. I think it's probably a good approach because you can do skin grafts as you can do some preclinical studies and with skin grafts from humans and understanding kind of how well things working. Obviously, it's going to be different when you're working on a live human versus a skin graft that you get donated to you. But I think it kind of de-risks the field in some ways, which I think would be good. And so I think I know the team well. I think they're very very good scientists, and we're rooting for their success.
And how do you envision the next generation of senolytics or broadly senotherapeutics? Are we still are moving into biologics?
Yeah.
Will small molecules still be the champions?
Yeah.
Maybe more
Uh,
gene and cell therapy approaches?
Yeah, I think it's going to be balanced between what is most effective, most safe, and then what is most cost compatible, right? So you think about some indications we're talking about, the payer tolerance for something like diabetes is not as high as it is for something like pulmonary fibrosis. So I think there's going to be kind of a Venn diagram between, what is the cost, how effective is it, how safe is it, how big is the need, and then where does that fit within the current therapeutic paradigm, right? So when we talk about treating diabetes with with senolytics or immune modulated senescence, it's not just about being good at senescence, it's about being good at the entire field. So like what is, think about standard of care and can you beat that with a senolytic therapy? So not just being the best senescence play, but being the best overall drug play. And so I think it's gonna be highly specific to which indication, what the payer tolerance is, what the tolerance is for side effects, and then what the current standard of care gives you or doesn't give you. So I think it's going to be very interesting to watch.
Do we need a companion biomarker for these kinds of studies?
I think it's a nice to have. I don't think you absolutely need it because ultimately what you care about is does it quantitatively improve a disease endpoint? And so if you have a companion biomarker for senescence, It would be great. Don't misinterpret what I'm saying. I'm just saying that the ultimate question though is if you remove senescent cells and you could show that, but then you didn't improve disease, then it'd be good to know that, but at the same time, ultimately what you care about is does it quantitatively improve endpoints in disease? I think that's the goal. The companion biomarker would give you a sense of maybe what percentage of senescent cells you need to remove in order to see disease improvements and how long it might take. So there's definitely value in it. I don't think it bottlenecks the field
Mhm.
entirely. I just think that it would be great to have it, but it's challenging for the reasons we mentioned. The way around it is to biopsy tissue and look at patient samples. Now it's hard because you might get sampling bias, um, or you might miss the kinetics might be different than what you expect. So, it's not a be all end all, but that's one way you can do it. You can biopsy, fat tissue from diabetic patients and see changes in senescent cells and then track that towards quantitative improvements in disease markers.
So perhaps more than a biomarker, what we need is to refine the classification of the different types of senescent cells, right?
Yeah, I think that's step one is to understand what is a pathological senescent cell versus one that's, senescent but not pathological.
I do see a scenario where you have different sub-classifications of senescence associated to damage
Yep.
specifically, and then program senescence that should happen, if you will. And actually, this brings me to another topic, the SenNet Consortium,
Yep.
which is an initiative led by the NIH to create an atlas of all the senescent cells in the human body throughout the lifetime. What will we learn in this atlas? What are you looking forward to?
Yeah, I mean, we've used this already, so it's been useful for us. I think what we'll learn is really how senescent cells behave in different disease states, right? The different organs, so I think our approach is likely to work. We've shown it works in pulmonary fibrosis and in diabetes. I think it's likely to work in more indications, but it's not going to work in every indication driven by senescence. And particularly the brain is probably unlikely that NKT cells have a role there. So, for example, if you really look at senescent cells from the brain, which would be senescent glial cells or senescent astrocytes, and you can see what's different about them versus their healthy counterparts, it'll give you clues on how to attack them. And I think that's the value. So, obviously analyze them and then figure out how to attack them is going to be a whole different, part of drug development, translation, etc. But it gives you a toolbox to say, okay, I can look at this senescent glial cell and understand how it's different than a healthy glial cell in the brain. And then from that, those clues, I can use those to then think about how I would remove them. Maybe it's xenomorphic, maybe it's a immune cell approach, maybe it's a skin-lick approach, see pros and cons of each. But it gives you a toolbox to work with to attack them.
And specifically for the research at your company, how did you use it? I'm assuming you looked at the senescent cells in the lung and asked the atlas, in which conditions do I get senescent cells in the lung?
Yeah.
Is that the way that you used it?
Yeah, we looked at the senescent cells from different disease tissue and we learned that what they're overexpressing on the surface that's would be attractive to the immune system versus healthy counterparts that don't express those. And so when you kind of say, okay, senescent cell expresses this, that can track to the immune system, and then normal cell don't express this, doesn't express any of these. So you can basically get a short list of things that senescent cells express that healthy cells don't. And then from there you can kind of pick through and say, okay, which one of these might signal an immune reaction versus not.
So you can basically design your flow cytometry gates using the information of the on the surface markers available in the SenNet Atlas.
Yeah.
Very, very, very cool. So Robin, um, I think we covered a lot of very interesting topics here.
Yeah.
Um, there is certainly a lot to discuss and a lot to look forward in the field. So my last question is unrelated to senescence. My last question is, what is the best advice you have ever received?
Yeah. I'll keep it to professional advice. I remember when I was working as an investor, as fresh out of school, and I was very, um, green-eyed and, didn't, was still becoming a professional. And I remember when I was working in Ohio, and it was like the fall and it was rainy, it was like 40 degrees, and I came in the office and there's kind of, like when you're living on the East Coast or the Midwest and there's bad weather, the city's kind of slowed down and people kind of like think about, and they spend a lot of time thinking or worrying about like things like clouds or like rain and like cold weather. And so I came into my office and I told my boss, and I looked a little bit, I think I looked a little slower that day. And he goes, "What's up with you?" And I go, "It's just rainy and kind of cold and miserable outside." And he goes, "You got to take days like this and make it an opportunity to work extra hard because everyone else is wasting their time thinking about what's going wrong. You need to think about what can go right." And so that was good advice. I think a lot of times we get sucked into negative cycles or what can go sideways, and as entrepreneurs, we always need to think about what we want to make go right. Said differently, don't predict the future, invent it. So, have a forward-looking, deterministic outlook on life is probably the best um, piece of advice I've ever had.
That's a very good piece of advice. Thank you
Yeah.
for sharing it, Robin.
Okay.
And thank you again for taking the time to speak with me
Yep.
and sharing what you're doing at the CTUs.
Yeah. Very good. Thank you, Jose. Very much appreciated.
Hi again. If you found this video interesting and would like more people to see this kind of content, consider clicking the like button and subscribing to our channel. Thank you for listening.